Hello, hello.
Not in Montreal, in Ottawa. Yeah.
Is this on? Yeah. So thank you for coming to the first colloquium of the year. So it's just to introduce myself. I'm Nico. I'm the chair of the colloquium committee. So if you have any complaints or suggestions, go talk to me. I think we have a full lineup. We might already have a cancellation, but as of today, the lineup is full. We're starting today with Professor Longtin. So thank you for coming. Come back every week.
Yeah, Nico has been great. So, well, I'm delighted to introduce Andre Longtin from University of Ottawa. He's a professor of physics there and the director of Ottawa's Center for Neural Dynamics and AI. In the way of background, he did his bachelor's and PhD in Montreal, Montreal, and then McGill, postdoc at Los Alamos, and has been at Ottawa since then. He has many recognitions. To name a few, he has been an APS fellow, got an NSERC Brockhaus Prize, and a Humboldt Medal. So with that, I think off to Andre.
Okay. Okay, thank you for the invitation. Can you hear me at the back there, everybody? Okay, great. I encourage you to ask questions as we go. If it gets overwhelming, we'll kind of cut it short. But I like it more interactive, especially as this is more kind of not mainstream physics, if I could say. So I'm gonna try and give you a picture of the kind of work that we're doing. And thanks, Sid, for inviting and having a wonderful visit so far. Okay, so physics of keyword is neural timekeeping and space mapping. So how do real neurons do things? This is what I'm interested in, been interested in this for most of my career. I'm a theorist. I did do some experiments while I was a PhD student at McGill, but since then I've relegated the hard work to colleagues who do the experiments. And I can just kind of freeload and go off and do theory with whoever I want. So it goes. So the roadmap for this talk is the following. So I'm going to give you some context because you don't really get this in an undergraduate degree. The neurophysics of perception and memory. Then I'm going to give you some illustrative examples of what I do. So some computation at the edge of chaos in the context of how do we focus on things. Speed invariant distance estimation, and this involves Hilbert transforms. 2D cognitive map. How do we build a map of space in our brains? And some new evidence, new experiments, and new data analysis that have led to some pretty critical findings in this area. And keeping time with adaptation, we're proposing a new method by which we can keep time in our brains. Finally, I'll say a word about, what we all like to do as physicists is inferring real physical models for things, but it gets very difficult. I mean, it's always difficult, but it gets difficult in particular with biological data because it seems that the nature has come up with many different solutions that work. How do you choose among them to find out which is the real one that works? Or maybe what ensemble of solutions work? So the kinds of questions I'm interested in are the following. So the context is the brain has billions of excitable cells called neurons, and they're supported by billions of other cells like astrocytes and blood vessels and so on. The neurons interact in various types of networks. So think of, networks of spins. This is the classic picture, but all kinds of networks. And their dynamics exhibit a mix of what I would call linearity and nonlinearity. H bar equals 0 in this talk. Okay, so we're not talking about quantum effects. Okay, it's good. And they're very noisy. They have a lot of stochastic inputs. We try to figure out what they're used for, the brain compensates for them, so on and so forth. So it raises the question of a deterministic versus probabilistic description of what's going on. So that means that you refer to tools from physics and frameworks such as a lot of the problems with neurons have to do with electricity, but also nonlinear dynamics because they have intrinsic nonlinearities, and they have they come in large collections. So there's statistical physics issues, and other parts of physics like neurophotonics come in as well for measurement technologies. The questions abound, especially about, and so there's lots of questions, but like, what are the levels of description you want? Like, what constitutes an interesting answer to these questions? So there's many different levels. I'm interested in the macroscopic, mesoscopic scale of description. So single neurons or collections of a few thousands of neurons. And that's pretty much the best observatory we have these days, especially with neuro-pixel silicon probes. They can give you a view of what thousands of neurons do at the same time. But then there's like a jump between that and whole brain recordings with the zillions of neurons, okay? So I'm in this micro mesoscopic scale, and I ask questions like, how do we keep time internally? What spatial coordinates do we use internally? How do we count? How do we store information? How do we recall information? And when I say, how do we count, you know, yeah, I can do 1 + 1 = 2 and so on. I can memorize a phone number in a book and go and write it over here, but what is going on really in the cells to do this? Okay, so that's the level of question. So this is my neurophysics agenda. So I'm going to talk about weakly electric fish, and I'm going to talk about mice. These are two kinds of animals that are beautiful models to answer some of these questions. And we hope that the questions are portable, you know, in a modular fashion to higher, you know, more evolved animals such as humans, but they are intrinsically interesting just per se, right? Just from a basic science standpoint. So this electric fish I was introduced to by my colleague, Len Mailer, who's a world expert on this, and been working with him for almost 30 years now at the University of Ottawa. So this fish is about this long. It acts like a swimming tuning fork. Those of you who know music, okay, like a tuning fork. They're the most accurate pacemakers in biology. They generate a field at a certain frequency, and that field is set up, as you can see, as a dipole. That's a snapshot at a given time, but it oscillates like at 1,000 hertz. So the polarity of the dipole changes. Okay, so they set up this active wave around them. When an object in the environment, such as that yellow dot there, yellow object, has a different conductivity than water, a different impedance, then it channels the field lines differently. In fact, the field generated by the fish induces a dipole in the object. The object becomes an oscillating dipole that is then imaged by sensors on the fish. So imagine it's like an electric retina that looks at oscillating dipoles, okay? So it's a beautiful system. And the cells that do this are they're almost identical to our auditory cells, turns out, okay? So we learn a lot about these guys, and we can port that to auditory research. They're extremely sensitive. They can go down to 1 microvolt, like below the noise floor. They can detect things that even our instruments can't detect. They're pretty amazing. They live in the Amazon, mostly in dark, murky water. They're active at night. Their eyes are not very useful for what I'm going to be talking about. And so this is the electric field, and when there's an object that goes by, it produces a modulation in the amplitude of their electric field. And this is basically the signal, the red signal that propagates to the brain. So that's this animal, that's the signal it gets. For mice, it's going to be another kind of signal. It's going to be searching around. It'll be a bit more intuitive what it's doing, but I wanted to emphasize this one. So for example, if you present a brass object to this fish, it'll produce an electric image that looks like a bump on the fish. And if you show it plastic, it'll be like a trough on the fish compared to the standard electric field it sees from its own wave, okay? And now we know a lot about the circuitry of these animals. So the sensors on the skin go through here and here and here and here and here, and they wind up in the memory. And we have lots of questions about this. And the timer I'm going to talk about a bit later this colloquium is basically here, which we think this is a timekeeper that records how the animal goes from one place to another and how much time it takes to go from one place to another. Would be this structure kind of embedded in here. We'll get back to that later. Here's the animal in question. Let's see if this works. I'm going to try. So it's going to be swimming around and it's looking for food. That's the electric field you're hearing. It's a pure tone. You saw that? Saw a little red dot at the end? That was a worm dropped into the tank and it swam by it, detected it. They're as good swimming forwards as they are swimming backwards, okay? They have a ribbon fin and it just went and ate it. So that was a pretty big signal. You can detect these, but they have to detect these within, like in muddy waters, plants and so on. So it's a sense that has evolved for millions of years to make these animals present today. I'm going to talk about, yes. So they can detect, so they actually have a second sense, okay, which yeah, so they have a, this is the electric active sense, and then they have a passive sense. If it's alive, it generates a little electric field as well, a little DC field, and has another detector that says, edible. So you can get back to that. So, but that was just detecting the location of it, and it can go and zoom in. Okay, so now, I said it doesn't need its eyes. So that means how can it see electrically around? But generally, when we have eyes, it's very difficult to understand how we see. Like we focus on things, we have a lens, but the lens kind of produces blur and corrects for blur somewhere in the back of our head here in the visual cortex. But the general question of focusing is all across biology. How do you focus on things? And this animal, turns out, uses a pretty fancy mechanism, we think, to focus on something. So what you can do is you can bring an object close to the animal. And when you do that, the electric field caused by the object goes up in strength. And then when it recedes away from the animal, the electric field drops like that. So you can look at one set of blue curves here. There's cells that look at positive contrast, other ones look at negative contrast, a bit like our own vision. Down here is the firing of the cell inside the brain of the fish, second order neuron, for those of you who know a bit of neuroscience. So it fires, and as the signal gets closer, what you notice is that there are more firings per unit time. This is not time, this is position, but it's related to time because we're moving the object in, and this is the position of the object. And so time is kind of implicit here. And then as it moves away, it pauses, and then it builds again some more activity. There's some red marks and some black marks. The red marks are like when the intervals between the firings of the neurons are very short. We call those bursts of activity. So the idea is that around this point, there's a big burst. It starts to burst a lot. 1.3 centimeters away from the fish. It goes from what we call tonic firing, or no pun intended, poisson firing, okay? And it's more like random firing that gets modulated in rate. And then once it reaches around this point, it starts to burst more. Okay, so I'm going to show you this is probably a focal point. This transition from tonic firing to burst firing, which is dynamically quite a big change because you start getting these very rapid bursts. We know a lot about the bursting mechanism in these cells from other studies. So what we can do is plot the distance, okay? So as the object comes in and it looms onto the fish, it's reducing the distance here as it gets closer and closer. And what you can do is something called a, other pun intended, not intended, the Fisher information, okay, from the statistician Fisher, right? So you can calculate, it's an information theoretic measure. It's a bit like a signal to noise ratio. And what you find is that the signal to noise ratio, as this thing moves in, goes up and goes down. You can estimate this, turns out, from spike trains. It's an old concept that's been used, it's all over engineering and so on, and communications engineering. So you get this thing right here, and Fisher information's maximum right there. And where is it? It's around 1.3, which is where we had this transition from tonic firing to more burst firing around 1.3. So around 1.3, system starts to burst, and there's a lot of information there. What that means is this is a point where if you move the object a bit, the response is strong. So this is a good place to have a focus, right? You don't want a place there, you can go move all over the place here and it doesn't change much the response. You wanted a critical point. And this is an example, and there are a number of examples in biology now, where the system is positioned near what's called a bifurcation, or a critical point, or a phase transition that has high susceptibility. You change the stimulus and you get a strong response. This seems to be the way this animal does this. Okay, so that's an example from sensory focusing. It turns out it's speed invariant. You can move the object at different velocities towards the fish, 0.5 meters per second up to 4 meters per second. You can calculate this burst fraction, how many bursts there are, and you see that they cross until a point where you get a lot of bursts. Forget about the x-axis here, but if you put all the speeds together, it doesn't care about the speed. It all crosses around the same point where it starts bursting. Whether the object is looming slowly or fast, doesn't matter. It just focuses the same way. So that raised another interesting question for us, is how do you get a neural system to actually not care about velocity, but care only about position? Because the velocity doesn't matter. Catherine, wait.
Because of burst, you've got like relaxation, so you're not going to get any more bursting for a while, so it becomes insensitive, the object.
So there's another question here, is like if it looks at something too long, it's like us. You don't see it anymore. So they're always trying to see things that move. Okay, so there's which is not part of the story right now, but it's in the equations actually. Let's go. Yeah. Okay, so here's an example. You know, this 1.3 turns out that it showed up in an experiment a long time ago by Walter Heiligenberg. What he did is he took these fish and he put them in an experiment where the fish is in the tank and has these bars next to it. And what they do is they move the bars over from one side to the other. So what does the fish do? If you move the bars over, the fish doesn't want to be hit by the bars, so it'll move with the bars. So you can see how well do they move with the bar. If you move the bar slowly or fast, you can get something called a transfer function. You can just see how well it responds as a function of the frequency. So you get all these fish out here who basically give up on the experiment and say, like, the hell with it, I'm not going to do this. And then, but you have all these fish here that actually, this is their gain, and it's again at 1.3. So 1.3 is like where they like to see the bars at a certain distance. It's their comfort zone. And it's also probably related to, they sway around in plants that move, slosh around in the Amazon. And you know, if you watch fish sloshing around in plants, they don't touch the plants, they move with everything, right? They keep a certain distance. So this is probably part of their focus. But I'm going to get back to this point about the speeds. How do you get something that's speed invariant? And so this is the firing rate of the cells is a function of time, but at different speeds, 0.5, 1, 2, so on. This is how the firing rate always goes up. And you notice that the rate goes up, but it doesn't actually, and then this is this 1.5 centimeter. If you look at the object position, it's coded by the firing rate, which is independent of the speed. The firing rate here is going to be independent of the speed. The object speed seems to be encoded in the rate of change of the firing rate. Here, hertz per second, so it's a rate of change of the rate. So speed does show up, it is encoded, but in the rate of change of the firing rate of the neuron. So how do you get this and this together? And I just give you an example from an old model we had, which is basic neural modeling 101, where you have a voltage variable, and the voltage has a natural RC time constant of decay. That's the neuron's RC properties. And it has a synaptic input. And it has this other thing, which is a fatigue property. So it's like W dot is minus omega over tau. This is like an exponential fatigue. The neurons fire too much, they have to slow down, basically. Okay. And if you look at this current here, what's happening in this experiment is that this synaptic current mimics what's coming onto the fish. It's the stimulus, and the stimulus is coming stronger and stronger. So this depends on time, but what we can try to show is how can it depend only on the position of the object, but not the speed of the object. And you can just fiddle with this pretty quickly. So you compute the net current into the neuron. A positive current promotes neural firing. X is the position. S is the resulting stimulation to the neuron. So you have S stimulation as a function of position, as a function of time. But you subtract off something which is proportional to the firing rate. It's subtracted from the stimulus because there's fatigue. If the neuron fires too much, there's fatigue. So a very simple picture is that you have to subtract off a quantity to this current, which depends on how much it's firing right now, how much it's been firing. And when you write this, usually as a convolution, it's the rate with some kind of response function of the kernel of the neuron. And this is an integral over time. And the idea is that you can change this to an integral over space because the object's just looming, right? And if you do the change of variables, you turn out to this thing becomes eta of x minus x prime divided by v. And this can be made independent of the velocity if, see, there's a velocity that comes out from a change of variables over here. It would be really cool if this velocity can cancel out with this velocity. Then it would be speed invariant. So how do you do that? It's very simple. Is you just make this kernel here be 1 over x, just 1 over x. If you do that, the velocity goes away. And that means that this kernel, in the language of theoretical neurophysics, is called power law adaptation. Power law adaptation is related to fractional derivatives, all kinds of interesting properties, math-wise. But basically, it's also what you do to do a Hilbert transform. So if you're doing a Hilbert transform on something, you render it velocity independent in this context. So the receptor is like doing a Hilbert transform. Yeah. I don't know. We haven't done the experiment, but I think it should. It's also like these fields propagate at the speed of light and water. So it's pretty quick. So I don't think it's going to matter much. But the acceleration is going to, yeah, I have to think about that. I mean, because there is acceleration in real life. But this transformation doesn't depend on the shape that I gave to x in here. X can be constant or can be changing with time. I mean, it could be a constant velocity or it could be accelerate. I don't think it'll change much. There might be another term that shows up in the change of variables. Interesting question. Yeah. What mechanistically is happening to create this in the cells? It's a certain class of currents that are in these neurons. They're called adaptation currents. They come in all kinds of varieties, and some of them have no intrinsic time scale. They're not exponential. but they seem to have these very long tail distributions, power law. So, and then that goes into the molecular biophysics, which is over my head, of how this actually happens. I don't think people actually know. It's an active area of research. All right, so now let's talk about cognitive maps. So we shift to mice. We leave the fish, okay? And this is the arena in which the fish, the mice wander. The mouse is right here. The arena is about 2 meters wide. There's holes all over the floor here where food will be hidden. There's a negative pressure, so the air kind of goes through the floor. So there's no smell cues. So the mouse, there's lots of things that have been done in here in this experiment. And this is experienced by Len Mailer and Kelly Shu, is a PhD student. And what you do is you can let the mouse in from different areas here, okay? And it has to find the food. And once it finds the food, it actually heads home very quickly. But finding the food is hard. So you can do experiments where you randomize the entrance every time. The mouse doesn't do very well in that case, right? You leave the food in the same place, but you make it come in from different random locations. None of us will be able to figure this out unless we have kind of external cues of the environment. Or you can keep the entrance static. I'm just going to talk about these experiments. And in particular, the mouse is building a map of its environment in its head of this 2D place, presumably. We all do this. And you know, the Nobel Prize was won a few years ago for our GPS, the internal GPS in mammals, right? So part of how this works. The question I'm going to talk about here is, can it do B minus A? Can you get the vector to go from B to A? The mouse is going to learn things here, food here. It's going to learn things here, that there's food here. And then, so here's the experiment. You give it food, and it has to eventually find the food here. And then you remove the food, and then it goes, where's my food? And it eventually finds another source of food right here. And then it gets better and better at finding this source of food. And then you remove that source of food. And then you ask, where is it going to go? So some do head home. Others actually do B minus A. An unrehearsed trajectory. Yeah? There's no olfactory cues. Yeah, because there's a negative pressure. The air gets sucked. And between every experiment, they clean the floor. And yeah. So maybe you want to help me. Let's just show a video of how this works. Yeah, go ahead. Those little circles, it's like they're doing hole checks. They're wondering, where's the food? They're looking at the holes. And now it's doing a trajectory from B to A. This is what's called a probe trial, when you remove the food at B. And now it says, okay, I'm not going to go home right away. I'm going to go straight to A. This is a pretty straight trajectory. Okay, so that gives you an idea. And then let's go back to the talk. All right. So this is what the trajectories look like. This is early learning. The mouse always starts here, and then there are two trajectories shown here, blue and red. The food is here. It's got to search quite a bit to find the food. It's early days. It doesn't know where the food is. It's just hunting around. Mice don't have a great visual system. They can't see very far. They have whiskers that are amazing. They have a close-up sense. And this is a late learning, and then it gets there pretty quickly. Notice that often there's trajectories along the walls. They just don't want to be eaten by an owl, right? They follow safe environments. So as it's learning, you can do heat maps of where it's visiting. The latency to get the food as a function of the learning trials goes down. It does this faster and faster. Its mean velocity of running around in here goes up with the trials, as you'd expect. The total distance traveled goes down. And then you can also do stuff on the hole checks. This is an active sensing animal, which I'm not going to talk about the active sensing part, but with its whiskers and with the hole checking, it's part of the equations of what's going on here. Okay, so I said I wouldn't talk about the random entrance. If you try this with random entrance, the mouse just can't figure out. This is where it estimates the target to be, and this is the food vector, and they're kind of uncorrelated. But with the static entrances, it kind of aligns the direction to the food and direction of where the target is from trial 1 to trial 14. You see it's doing better. And so there's this target estimation vector we came up with. So this is with my postdoc, Mauricio Girardi-Chapo, who's now a physics prof at Santa Catarina in Brazil. He looked at the trajectories here, and we're basically kind of put a lattice inside the arena. And at every point of the lattice, you want to know what's the probability to go left, right, up, down. And you estimate that from the data. And for example, this would be a place where, you see a lot of the trajectories come through this 3-3 point. So there's a map at 3-3, which is like a narrow that goes straight up, because most of the trajectories just funnel through there. And you do this for the whole environment. So you can construct these displacement maps with the horizontal one going left and right, vertical one up and down. The probabilities have to sum to 1. And then you can look at, I won't go into the details here, but there's a baseline property or baseline probability to move anywhere. The problem is sometimes the animal doesn't move. It doesn't go anywhere. It's like your particle just stays on the lattice and just doesn't jump, stays there. And you can cook up these transition probabilities. S is the number of times it visits various cells. And in the end, you can find that you can explain these probe trials because the animal has been able to build a map of the environment and know that there's this connection between the two to do that subtraction. So it took basically a shortcut, right? And it's basically doing this. So, and what's building up in the neural circuitry is something like this. Like you have an environment with all these arrows, and then you're slowly funneling the arrows down to an environment. And so what we're working on now is how you get these transitions from cell to cell with some kind of probability of jumping from one place to another. How is it learning? Where is it going? Is it learning from the beginning towards the end? Is it learning from the food site going backwards? How is the learning actually occurring? How are you changing the transition probability on this lattice is what we're currently trying to figure out. And to get these trajectories that are eventually, at run 1, they're all over the place. At run 50, they kind of go ballistic to target. Okay. And so I'm going to spend just a couple of minutes talking about this time adaptation now. So time adaptation, keeping time with adaptation is, again, let's see if this works. this is with these people. Len is my longtime collaborator down here. And the experiment's done by Avner. And Raphael is a theorist working on this for his PhD. So here's the experiment. This animal is looking for food. Let me go back. That's another annoying sound. It's a different kind of fish. But what you see down here is the sampling rate of its environment. It's actively sampling its environment. What you're going to see is it's encountering different objects. And it learned that there was food, I think, over here. But now it's going to come here and there is going to be no food. And it's going to look around. This sound you're hearing is how it's actively sampling its environment. And basically how pissed off it is when there's no food. You'll hear what happens. Okay, let's try this again. See, like, where's my food? All this red code right here, right? So we're back to a fish, by the way. We're no longer talking about mice. Okay, so that much is clear. And but it's learned that the food is there, but there's different obstacles, there's different landmarks, and that's what we all do when we're learning where things are. We know, it's this distance to here, and then you turn and go there, and there's times between encounters. And what I'll try to explain is that there's a speedometer that all fish have that are basically pressure detectors on their body. Okay, it's called the lateral line system. That's the speedometer. And the timer is the adaptation. So if you have the distance traveled and the time between the encounters of interest, then you can just multiply the two. The distance between object 1 and 2 is the mean travel speed times the elapsed time, right? So simple picture. And this is what you do in the experiment is you bring the object close to the fish and then you move away and you wait a certain time and you bring close and you move away and you bring close. And what you see here are responses of the neurons. The intervals are random in length and duration. So because there's fatigue in any neuron, if it fires a lot and it gets stimulated again not too long after, it's not ready to fire a lot. That's what happens on this third response. It had a good response here, a good response here. The second one came with a shorter interval than compared to that. It's a smaller response. So from the inside of the brain, no one's telling the fish where things are. The fish has to figure that out. So if you're inside the brain, what we would call the homunculus for humans, or the fish unculus for the fish, it has to look at these responses and decide what was the interval between these places that I'm trying to map in my environment, right? So turns out that if you plot the responses here, then and you relate them to the interval, there's this sigmoidal function. So there, if you get a certain response, you can look back and you can measure the time. So if the circuitry is able to do this, it can use the responses and learn that this response means this time interval. Because there's like a bijection between the two, right? So, but that's based only on the most recent interval. So some cells actually care about many intervals. These cells care only about the most recent interval. So you can play around with this and make mathematical models on and do maximum likelihood estimation, where you have some internal latent dynamics inside the neuron. And for those of you doing AI or machine learning, you'll see some of the terms being quite similar here, right? Latent dynamics, a ReLU function here for your neuron, and this part they don't throw in too much, but it's like a Poisson distribution of spikes. You can explain what's going on with this model. You can actually show that about 500 cells are necessary to just do this job, to estimate where things are. The question is now, what happens when there's multiple intervals? Then these cells will not. Yep. You have to just in parallel for now, just parallel. They're averaging out the noise. Yep. And then what you're basically doing is like, what's the likelihood of these intervals based on the responses? And you can invert Bayes' rule. This is going to be the probability of the responses given the intervals. And then you can do your tricks with this model and write out the math and then find what the estimate is. It turns out it's a lot more difficult when there's two intervals, because what it turns out, I don't have time to show you the theory, it's a bit hairy. If you want to estimate two intervals into the past, you actually can't do it if all your neurons are the same. You have to have two populations that are different. One population has 5,000 cells and they have like no memory. The other population has 5,000 cells with some memory. This is not the only combination that works. So there you can estimate very well the last interval, but you can't estimate, you can still estimate the previous interval. But realize this is going to go down exponentially fast. You won't be able to estimate much further into the past. It's just too long. But these are memory processes on orders of seconds to almost a minute that we're looking at with this kind of dynamics. Okay, so in all this, we're trying to infer physical models. And it turns out it's quite difficult to get one good physical model for something. There's this problem of degeneracy. So for example, that memory circuit that I talked about that the mouse is using to figure out where things are in space, this is the circuit in a very simplified form. It's actually much more complicated than this. We're currently putting electrodes in the brain and measuring that while the animal is searching to decode the neural language. How are the neurons doing it? But models of this kind are just crazy complicated. And Alex here with my colleague Jacob has been working on how to estimate models, good models. And basically, this is the kind of thing that you want to do. And I'm going to cut it short here, but you want to estimate some parameter in a model, and you can do this with machine learning techniques. You can do this with maximum likelihood techniques. You can do, all your favorite Monte Carlo, Hamiltonian, there's lots of tricks to do this. But often what happens is that the energy functions or the energy landscapes have multiple minima, right? Those who do protein folding, in the biophysics realm, know about this. There's like minima all over the place. So here are three parameters you're trying to estimate in some model. You can do pretty well. You can look at this model here, which has like 4 areas of the brain, and there's connections between the different areas. You can these days record the activity of all these, and there's a bunch of parameters you can try and estimate. And but at the cost where you're saying this is the best likelihood for each of the parameters, but look that there's a lot of different trajectories that are close by. How do you know that's really the best one? So this is a very big challenge in understanding what kinds of models can reproduce the data. And Alex has just put out his last paper where basically he's able to generate what's called not just the maximum likelihood, not just the one that has the best likelihood, but a metric that can say, how much are you willing to bet on this? You need the distribution of your risk around each. And then you can say, okay, these models, have an expected risk smaller. These ones are higher risk. I'm going to throw out those models, but these ones are both very good. Which one do you pick? Well, there's a bootstrap method that I won't get into that you can estimate a distribution of the risk. It has to do with kind of simulating the cumulative response, cumulative probability function for your risk in your system, and then you can choose the one that has. Then you have two distributions to work with. It's much easier to bet on something when you know the distribution. And this we're importing now into this machine learning. In our paper, we illustrated it with the ultraviolet catastrophe, but also with a neural model, to say this is how the risk can help you understand when you include amounts of uncertainty about the experiment, you can generate these risk distributions. Okay, so I've shown you about focusing system position at the edge of chaos to detect changes, speed invariant coding using power law adaptation or fatigue. Keeping time with adaptation, you need many populations with different parameters to infer sequence of times. The hippocampus can do vector subtraction to infer shortcuts. And this is evidence, this is the first evidence of actually a real 2D map actually happening in an animal. The frontier is to record from the brain at the same time as this behavior is happening to understand the dynamics governing the formation and the operation of this map. That's my hang up, trying to understand what the neurons are actually doing. But it's the big team efforts to understand the whole process of spatial memory. And with that, I will thank you for your attention. Yeah, hi.
Maps? For the mice maps, are the transition probabilities local, like they depend only on the nearest neighbor? Is that your assumption?
That is the assumption so far. But it might be that the mouse is making some average over different cells, looking back, non-nearest interactions just with some decay going further away. This will probably all smooth things out, but we don't quite know how the plasticity rule operates. That's what we're trying to figure out. But the recordings from the cells are probably going to help us to do that.
So since this is the start of a new term, I'll say probably all of us are worried not only about how we learn things, but how we forget them or how quickly we forget them. So I gather that after a long period of time or after being exposed to a lot of different stimuli, the mice will eventually forget their maps. Do you have any insight into how that process works? How the maps get overwritten by other maps or maybe just disintegrate?
Yeah, so it's an excellent question. We have, there's a lot of research going on to that. First of all, there's two parts to your question. Multiple maps, because you can be doing this A-B thing part of your day, but you're also doing other things part of your day. So these maps are co-localized. There's remapping going on various time scales. So that's one thing. But the general thing about plasticity of neurons, it seems like neurons are hardwired to go back to where they were. It's called homeostatic plasticity. And there's a time scale to that, which people are trying to figure out. And what's the mechanism? What's the good trade-off? Because if you don't forget anything, after a while, you can't learn anything, right? So you have to be able to forget some things to make room. Active forgetting is a very big, not just in my field, actually in what Sid's doing and with genetic networks is also a big thing. Yeah.
So can we understand the need for two different populations for the two different time intervals is you need something to break time translation invariance?
So sorry.
You need something to break time translation invariance in order to like measure two intervals. So you need something with power law decay and something with an exponential time constant.
So that story did not really bring in the, so there's adaptation going on, right? If there were no adaptation in that cell, if there's no fatigue, then you wouldn't have this coding even possible. But the invariance, so you're talking about translation and time translation invariance would mean like to repeat the same experiment like 10 seconds later? Or what would be the particular question?
One populate. If you only have the population with the power law response, then that's like, sorry, like the exponential response population has a time constant. So that fixes a time scale in the system where the other one doesn't. And so that, yeah.
So the question, this necessity of heterogeneity to memorize more than one interval in a sequence doesn't really depend on whether it's exponential or power law.
Okay.
What you can do is you can do a Fisher information calculation. You get this Fisher information matrix, and you see that to make that invertible, you need two populations at least. You need as many populations as there are intervals. Yeah.
Maybe one last question.
Thank you for a wonderful talk. I was just wondering, the A minus B experiment, that's a two-dimensional problem. The mouse, what do you think would the implication be in 3-dimension? Do you think it makes?
For flying mice.
Yeah.
Yeah.
Or for birds, or for your fish that has to float in the Amazon?
Yeah, the experiments that we did with the fish are actually in shallow water to prevent that precise problem. But there's a lot of work being done on bats, and bats fly around and they have the transmitters on their head while their neurons are being recorded from, and you can, a lot of what we're talking about is also seen in bats, place cells, boundary cells, vector cells. And so in terms of people didn't have a clear idea that a 2D map could even exist. They all kind of assumed it, but they never actually saw it. So this adds fodder to the possibility that 3D maps exist, but they're much more complicated.
And maybe one last question. I know people are, there's a class clamoring outside, I think, but
I wanted to wonder, so if I understood correctly, this power law adaptation is needed if you want to extract a position from this information.
The Hilbert transform is needed to extract position.
To extract the position.
Yeah.
But the fish might also want to know at what speed something is. So you might want also to have similar mechanism to extract the velocity. Or
the velocity seems to be okay just by looking at the rate of change of the firing rate. Rate of change of the firing rate is enough. But the ambiguity but to make the position, estimating the position independent of the velocity, that you require a Hilbert transform. Yeah.
Great. Before everyone vanishes, we should thank Andre one more time.